Okay, we better get started. We have a lot to cover today. We're going to transition. Last time we talked about the human reference genome. Now we're going to talk about one of the fundamental reasons that the reference genome was created. That is to study genetic variation in the human population. Before I get into that lecture, I just want to highlight a few slides that we didn't get to last time that are relevant, I think, to maybe one or two questions in your homework. Relates to two Unix commands, sort, which, as you might guess by the name, it actually sorts a data file in either alphabetical or in numerical order. And this other command called unique, which basically in sorted groups of data finds unique values in those sorted groups. So for instance, in this block of text, there's the number 210.201:A multiple times. The unique command, if this data is sorted this way, will tell you, yes, this is the unique value in that block. Okay, there's a lot on the internet about sort and unique. Combining sort and unique is a common pattern for data analysis. It's relevant to what you're doing in the homework. So look at slides like 40 through 47. There's probably a million web pages about this same thing too. But just a tip for the homework that's due Thursday. Okay, so moving on to genetic variation. I think pretty much everyone here, I think, at least has some sense of what I'm talking about here. This is differences in the DNA sequence between individuals within a given species population. So if you looked at all the DNA for every fruit fly in the world, they all have the same basic DNA sequence, but there's differences, right? And in the human population, the main area of interest is what is the connection between those DNA differences between each of us in this room or worldwide and the phenotypic diversity that exists among all of us here. Not even talking about disease risk or anything, just traits, eye color, hair color, stature, weight, predisposition to type 1 diabetes, etc. And so this study of genetic variation is really fundamental to the explosion in genomics, human genomics. It's a big area, big focus of research here at the U and at pretty much every university in the country. Okay, so what is genetic variation? We talked about that briefly. I mean, if you compare any two individuals, human individuals, our DNA, and we learned last time that the human reference genome is about 3.1 billion base pairs. We inherit two of those haploid genomes, one from mom, one from dad. And if we compared any two haploid genomes between any two individuals, the DNA bases in those 3.1 billion base pairs that are being compared are like 99.8, 99.7. These numbers are adjusted slightly from year to year. Identical. So very, very small percentage of those 3.1 billion base pairs are different among individuals. And that works out to an average of about one difference. If we started at the beginning of chromosome 1 and just marched along and compared those two sequences, we'd see on average a difference between 1 out of every 620 to 1 out of 750 base pairs. And I'll talk about why there's a range later on in the lecture. That's quite different than Drosophila, for instance, which has a much larger population size. You know, there's something like 8 or 9 billion humans on Earth. There's probably 8 or 9 billion Drosophila just in a county in North Carolina, probably. Now, if you compare two different primates, say chimp and human, it's pretty similar. I mean, the DNA, even at that level, is quite identical. 99% of bases, if you compared the human chromosome 1 to its homologue in the chimp genome, which I think is probably chromosome 1 in the chimp genome, we'd only see one difference out of every 100 base pairs. Okay? So while that seems like a very high degree of identity, one of the things to recognize is that the human genome, remember, is massive. So even though it's 99.8% identical, if you do the math, 0.2% times 3 billion is a lot of differences. And it turns out that that's millions, something like 4 million base pairs that will differ between any two individuals. And those differences are going to be at different locations depending upon which two individuals you're comparing. And that's, if you think about the combinatorics, those 4 million differences are more than enough to explain all the different phenotypic diversity that exists on Earth. Okay, so getting back to really what we're talking about, these are differences at a high level in the DNA content. So whether you're an A and I'm a T, or the structure. So we hopefully know that chromosomes can differ in structure. So there's aneuploidy. So anyone heard of a trisomy? Trisomy 21. That's a third. Those individuals have a third chromosome of third instance of chromosome 21. So if you liken the human genome to like the book Moby Dick, some individuals might have duplicates of chapter 7, or some people might have one paragraph in chapter 8 completely twisted around and inverted. But most of the DNA differences are spelling mistakes in that book, so nucleotide differences. So this is updated to be 99.5%. It says 99.8%. Seems like I updated it based upon a recent paper there, but didn't update it here. So this is depending upon how many new individuals are being studied in these ever larger DNA sequencing studies. These estimates get revised from time to time. Human genome's big, so there's a lot of opportunity for genetic variation. And among all the individuals who have been sequenced so far, I don't know why this keeps happening. We know of more than 100 million genetic differences that exist within the human population. The jargon is that those variants are segregating in the human population. And I think if we once we get to sequencing on the order of million or 10 million or even 100 million human genomes, this number is going to just keep going up and up and up. And that allows for essentially an infinite number of combinations of alleles. And the details about exactly which genetic variants we inherit from our parents, that explains why, mostly, as Galton showed, our height is roughly an average of the heights of our mom and dad, and that comes from the fact that we're inheriting about 50% of DNA from mom and dad, and they vary risk for different disease phenotypes. Bless you. Okay, so really what we're doing is searching this massive genome, which is these green letters. Most of the time, if we compare two reference genome or two individuals' genomes, the nucleotide sequence is the same as in green here, but sometimes there's these breadcrumbs of genetic variants littered throughout the genome, and really that's what we're looking for. The areas where any two individuals' genomes are the same, in many cases, we don't care about those. If we're trying to understand the relationship between a genotype, the DNA sequences that an individual inherited, and their phenotype, we're really caring about the differences. Okay, so there's three primary types of genetic variation. I'm going to ignore a couple minor types, but typically we're talking about these things called single nucleotide polymorphisms. So poly, many, morph, states. They have multiple states. So in the human population, sometimes we see chromosomes where it's a C here. Sometimes we see individuals with a T at that site. And most of the genetic variation that we're aware of in the human genome is this type, these spelling differences or single nucleotide changes. These are sometimes called single nucleotide variants, SNVs. Sometimes they're called SNPs. Sometimes they're called polymorphisms. There's a lot of different jargon. That's an unfortunate thing in genetics. We also have these insertion or deletion polymorphisms. So again, polymorphisms, multiple states. Here the two observed states are this individual chromosome has a T and G here, whereas that T and G is deleted in this individual. So it's an insertion dash deletion, insertion or deletion, because it depends upon which individual we're talking about. With respect to this individual, this is an insertion. with respect to this individual, this is a deletion. So it's sort of symmetrical. That's why it's an indel, insertion or deletion. Now, the third type, which we'll talk more about in a later lecture, are structural variants. These are situations where there's much larger insertions or deletions, sometimes megabases, that differ. So a whole chunk of a chromosome may be deleted in an individual. And it used to be thought that these large-scale structural changes, like deleting chapter 7 from some individual's genome, was sort of restricted to tumor genomes. We've known for a long time that solid tumors have these really, really destroyed genomes, often large duplications and deletions and inversions all over the place. But what we've realized over the last 15 years is that these are actually quite common in the genomes that we inherit from our mom and dad. So every one of us in this room have something like 5,000 to 10,000 of these much larger insertions and deletions. And looking around the room, it seems like every one of us is fine, despite having all these deletions and duplications, right? Okay, so there was a project that completed a couple years ago called the 1,000 Genomes Project. Despite the name, they ended up sequencing 2,504 genomes. And the real goal of this project was essentially to catalog all of the common genetic variation that exists in the human population. And one of the other advances of this study is that prior to it, most of the genetic variation that we knew about was ascertained from people of European ancestry. So a primary goal of this project was to actually study individuals from different world populations and get a sense of how variable genomes are at the DNA level in people of European ancestry or Asian ancestry or African ancestry, etc. And I just highlighted in green some of the salient points from the abstract. I think this sentence is a beautiful abstract sentence. It basically summarizes all the results. In many ways, you could sort of read this and be like, okay, I'm done with this paper. Care about the details, look into the figures and the methods. But as I mentioned before, a typical human genome differs from the reference genome, what we looked at last time, at about 4 to 5 million sites. While most of these variants are these SNPs and indels, the first two categories, they find that a typical genome contains 2,000 to 2,500 large deletions, copy number changes, these retrotransposon insertions that we talked about, et cetera, et cetera. This number is actually now been proven to be quite an underestimate. It might be something like 10 to 20,000. And the reason it's an underestimate has to do mainly with the sequencing technology that was used to study these 2,504 genomes. Some of the more advanced emerging DNA sequencing technologies are better suited to find these larger scale structural changes. This gives us the number that I mentioned on the previous 2 slides ago, sort of the average density of these genetic differences in the human genome. That's a measure, population genetic measure that's called pi, or nucleotide diversity. So how much diversity is there in a species? Okay, why do we care about this stuff? I think it's probably rather obvious, but there are many different reasons that we care about knowing what genetic variation exists in the human genome. Primarily, this is to understand the relationship between genotype and phenotype, or the relationship between genetic variation and traits or disease phenotypes. And for a long time, the way this was done was by so-called cases versus control studies, where let's say we collect a cohort of, 10,000 individuals with type 1 diabetes. We identify ideally, an equal-sized cohort of controls, that is, individuals that are age-matched, ancestry-matched, sex-matched, and don't have type 1 diabetes. And then essentially what you're doing is studying all the genetic variation that exists in the group with disease, all the genetic variation in the group without disease, and then essentially applying statistics to figure out which of the genetic variants are most strongly associated with individuals with the disease. By and large, those studies have been successful. I will counter the claim that these so-called genome-wide association studies have been unsuccessful. they have been very successful at identifying loci that are very, very correlated with the disease phenotype. However, they don't get at mechanism. They don't tell us why they're associated. And that's what sort of, a lot of biologists sort of poo-poo these studies because we don't get mechanism, but you can't get mechanism unless you know where to look, where the associations are, and that was the whole point of these studies. Also, so this is kind of a study designed for common disease, but if we're studying rare disease, such as this autosomal recessive cartoon, we're really looking for situations where mom and dad are, say, heterozygous for a given variant, and then we're looking at the segregation of all the variation in that family with individuals that have disease, I guess is this gray color, versus those that are unaffected. And this is a big strength here at the University of Utah. We have an undiagnosed disease network clinic. Probably maybe heard of the Penelope Project. The center that I participate in, that's a big focus. We study genomes of families that have rare disease and are basically finding the genetic variation that are in those parents and kids, and then basically running algorithms to find genetic variants in those families that are most likely to be connected to the disease phenotype in the kids. Okay, so that's sort of the NIH focus. The NSF focus would be studying how genomes evolve and species evolve, and these genetic variants essentially are breadcrumbs that we can use to sort of reconstruct the evolutionary history of a species or a group of species. And that's using essentially methods that akin to phylogenetic methods, if you've used anything like that. We're not going to talk too much detail about that, but it's hopefully clear that using genetic variation collected from millions of individuals might be able to reconstruct sort of the evolutionary history of our species and the species of other primates and other organisms. Okay, so I want to get a bit into some nomenclature, and I emphasize this bit because this is terminology that is conflated all the time. I hear it in lectures at our university all the time by card-carrying geneticists. It's something a process and a fundamental difference that we really need to understand. A polymorphism, a variant that is observed among many individuals in a room or in a worldwide in a species, is not the same as a mutation. Not all mutations become polymorphisms, right? Because when mom or dad make a sperm or egg, those haploid genomes sometimes have new mutations in them. That's a consequence of replication error during meiosis. And in the case of dad, it's more obvious. Lots of sperm are generated. Only a small minority of them ever go on to fertilize an egg and lead to an individual. So there's lots of mutations that arise in a sperm lineage, or an egg lineage for that matter, that never go on to fertilize an egg and therefore have no opportunity to become a polymorphism by being transmitted to the next generation and the next generation. So it's one of these things where we really need to be careful about terminology and be using the right word for the mechanism or the type of thing that we're trying to find. So, or we're talking about rather. So let's imagine, I think this is probably clear, but let's just walk through it. So let's imagine we have a toy population of 10 identical chromosomes. So let's imagine these are snippets of DNA from 10 sperm at the same locus. They're all identical, right? And that makes sense. This is the DNA sequence of the man who generated these gametes. Okay. Now let's imagine one of these gametes is different. A mutation arose on this gamete that is different, makes it different from the other 9 gametes. So this mutation is private to this chromosome, or this individual is probably the wrong word here, this gamete. Okay, so if this gamete or this sperm does not go on to fertilize an egg, that mutation this special T mutation disappears until it arises again. That mutation may arise thousands and thousands of times among a sperm population, but never, ever be passed on to the next generation because it never fertilized an egg. What if it did? Well then, if that mutation is on a sperm that goes on to fertilize an egg, that zygote goes on to produce an offspring. That offspring then that mutation is now technically not a mutation anymore. It was a mutation, now it's a polymorphism. There is an individual in the human population that's walking around that has that variant. The frequency of that polymorphism is 1 chromosome over 2 times the number of people on Earth. Imagine that's the first time that mutation ever arose, right? Because if it arose on dad's sperm, went to fertilize an egg to become a zygote, then it's on 1 of the 2 chromosomes in that offspring. So the frequency of that polymorphism is 1 over 2 times the population size, human population size. If that kid goes on to have offspring, and those offspring, let's say this is the kid, that kid grows up, has kids, passes that now polymorphism on, the frequency has increased, right? There are several offspring that have that polymorphism. Okay, so the frequency of a polymorphism is a function of how many individuals have inherited that variant that originally arose as a mutation way back when. Okay, so the basic disconnect is mutations, not all mutations become polymorphisms, but all polymorphisms were at one time new mutations. Yeah?
Does it have to be in the germline then to be a polymorphism?
Yes, because if there's, I didn't really wear sunscreen until I was about 35, so there were lots of mutations in all my skin cells, but they have no opportunity to go on to become polymorphisms because they're not in the germline.
But if multiple people had the same mutation, it would still just be a mutation.
If multiple people had the same mutation and didn't pass it on, it would be a mutation. If multiple people had the same mutation and passed it on, it then becomes a polymorphism, but the origin of that polymorphism becomes unclear. And that happens because there are parts of the genome that are hypermutable. And so if it's a hypermutable region of the genome, it's more likely that many individuals will have that same mutation arise in their gametes, and that mutation will arise independently and become polymorphisms independently. What we don't get for free if we sequenced everyone's DNA in this room and found all the polymorphisms, sites in our genome where at least one individual had a change, we wouldn't actually know the mutational history directly. We would actually have to do something far fancier to figure out whether that polymorphism arose from a single mutation a long time ago, and that's what explains its frequency, or if it arose independently in multiple different individuals more recently. Okay, so we talked about this maybe 10 times now, or at least in 2 different lectures. Humans are diploid. We inherit 2 sets of 23 chromosomes, 22 autosomes from mom, 22 autosomes from dad, and then sex chromosomes that explains their own sex. Those are haplotypes. Together they form a genotype. And as we can see, the 2 haplotypes, if we zoom in on this part of this chromosome, the 2 haplotypes that we inherited from dad and mom are by and large identical, but here's a site where they differ, and that makes us, this individual, this offspring, heterozygous, inherited an A from mom and a T from dad. Together, that makes our genotype. So the genotype of this individual at this site would be AA, TT, CC, GG, 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 TA. Okay. So this is based. Yes. Yeah.
To rewind a little bit to when you said like some parts of the genome are highly mutable relative to others.
Yeah.
Is that simply due to selection pressure? Like they don't really contribute to like important processes, or like are they physically and mechanistically like easier to mutate?
Both. So some regions of the genome that have less genetic variation are under constraint. That is, they're less tolerant of mutation because they're essential to the development of the organism. On the other end of the spectrum, there are regions of the genome that are hypermutable for possibly two different reasons. One, biochemistry. We talked about CPG dinucleotides, so C followed by G, those cytosines become methylated, and that's a biochemical explanation for why those Cs are hypermutable. They often become Ts by a process called deamination. But also, regions may have a higher level of mutation simply because it doesn't really matter what the sequence is there. There's less selective pressure on those regions. And teasing apart the mechanisms that underlie why some regions are more or less mutable is something that a lot of labs work on, including my own. It's a big open problem and question in genetics. Okay, so this is essentially Punnett square stuff all the way back to, for me, 9th grade, probably for you guys like 5th grade or something. But so let's imagine these two individuals were heterozygous, and we're tracking the outcome of offspring of these two individuals based upon what they inherit at this site. So obviously if they inherited, I can't reach that high, if this child inherited that gamete from dad and that gamete from mom, this daughter would be homozygous at that site. So same, the zygote has the same two gametes. In this, there are two possible ways where offspring could be heterozygous. Situation where this daughter inherits, say, C from dad, T from mom, or C from mom, T from dad, right? And then the third case, child is homozygous, they inherit the other two gametes. Okay, so let's just look at data. We've been looking at cartoons so far. This probably doesn't render too well in the back, but in this case, we're looking at a heterozygous site. Let's imagine this is a program called IGV. We'll talk a little bit about this later. This is DNA sequencing data, let's say from this daughter. Okay, we have to pretend a little bit because the alleles here is a G and an A, but that's the complement of C and T. So what we can see at each of these gray bars is a DNA sequencing read from an Illumina sequencing project. And what IGV does is whenever the read has the same allele as the reference genome, which is on the bottom here, it keeps the color gray. But whenever the DNA sequencing read has a different allele than the reference genome, it colors it. And so what you can see is that it's roughly 50-50 reads that have A's, these gray ones, and reads that have G. And if an individual is truly heterozygous, we would expect a roughly 50-50 ratio of DNA sequencing reads at a particular site at which an individual is heterozygous to be one allele, and the other 50% of the reads to be the other allele. We'll talk in 2 lectures about sort of exceptions to that and some of the math and stats behind why and when that works. But conceptually, if the fertilized egg was heterozygous, that means every single cell in the fully developed organism will also be heterozygous. So if we drew blood from daughter, isolated white blood cells, randomly sampled DNA sequencing reads from the millions of cells that we drew, and basically align them to the reference genome, we should see a roughly 50-50 ratio of the two alleles that daughter inherited from her parents. Yeah?
How does the reference genome handle variation within itself between the two haplotypes?
Generally well, but sometimes poorly, and we'll talk more about that. So there are some biases. The reference genome is like the jigsaw puzzle that we use to figure out where to place these DNA sequencing reads, but that leads to some problems. There are parts of the genome that are so repetitive or so structurally variable that it's very difficult to align DNA sequencing reads to them. and therefore we have a little bit of blinders in terms of genetic variation that exists in those parts of the genome.
And would we have a clear like sequence on the bottom if the two haplotypes of the reference genome were different?
So that's, misconception. Reference genome is just one haplotype.
Oh.
It is a consensus sequence, and so we're using that as a guide to figure out what haplotypes are in a newly sequenced individual, but there's only one, well, until a couple weeks ago, there's really only one sequence for the reference genome. There's projects now to represent the reference genome as sort of a collection of different sequences, but no one's really doing that now yet. Okay, so let's talk again about the birth of a new variation. So sort of trying to ram this home. Let's say mom and dad are homozygous at this site of interest that's colored. They both inherited a C from their parents. But let's imagine that a new mutation occurs in, let's say, father's sperm cell. That sperm is the one that goes on to fertilize an egg that has either these two haplotypes, one of these two haplotypes from mom. And now that zygote goes on to form their daughter. This individual is now heterozygous, and we know that this is a de novo mutation because we can look back. If we had DNA from mom and dad and kid, we'd realize that this allele, this new T allele that arose on dad's sperm, could not exist without mutation because mom and dad were both homozygous. With one exception, sequencing technologies unfortunately aren't perfect. So this also could be just a sequencing error. So a lot of the work in finding mutations in cancer genomes or in children's genomes that have developmental disorders is trying to separate out true spontaneous mutation in the sequencing data from DNA sequencing errors. Thankfully, that problem is a lot easier now than it was 10 years ago, but some of the first papers using exome and genome sequencing to find de novo mutation, they spent like half of their methods section talking about how they figured this out. Okay, so I think it's worth thinking a little bit about how frequently de novo mutations occur. So these are de novo mutations in the germline. Work in my lab and many other labs over the last 10 years have shown that essentially the human mutation rate is about 1.1 times 10 to the negative 8th per base pair per generation. So that might sound a little bit cryptic, but so that's a whole genome, 3.1 billion base pairs. The rate is 1.1 times 10 to the negative 8th. So the genome is 3.1 times 10 to the 9th, 3 billion. So you multiply 3.1 times 10 to the 9th by 1.1 times 10 to the negative 8th, and you get about 30 to 40. This is just factoring out 10 to the negative 8th and 10 to the 9th, just adding exponents. And you end up with an estimate of about 30 to 40 new mutations per haploid genome in the germline. There's 30 to 40 per mom's gamete, 30 to 40 per dad's. It's actually more on dad's than mom's. But the net effect is that we get around 70 to 80 new mutations in every offspring. So each of you, on average, differ from your parents' DNA at about 80 positions in the genome. Okay? The question of what the human mutation rate is a bit of a circular question because we think of this as an average rate, but we just talked 5 minutes ago about the fact that some parts of the genome are really mutable, some parts of the genome are not so mutable, and so this is an average, but it doesn't really apply. That rate doesn't apply to every part of the genome. So parts of the genome that encode protein coding genes probably have a lower mutation rate than parts of the genome that don't encode protein coding genes because they're less important to the development of the organism, so they're more important to the development of the organism, so they're under greater pressure. Yeah.
Is there very much research into like variation in the number of de novo mutations between populations?
Of the same species?
Yeah, like in humans.
Lots.
Yeah.
Tons of papers on that, yeah. So mutational processes are about the same, but there are different types of mutations that are enriched in different subpopulations, and why that is sort of an open question. Families vary in the amount of mutation that arises in their germline. We have a paper on that from this year. Yeah, it's variable. So that's another reason why this sort of average rate is a bit flawed. We need to be thinking about rates as a function of ancestry, age of the parent at the time that the child was conceived, all these different things.
There's probably a lot of like environmental factors.
Yep, exogenous mutagens. We all, well, not all of us, but many of us do bad things, drink and smoke and vape and do all these things, and all three of those things and many others are germline mutagens.
So, Aaron,
Yeah.
so this is all the human mutation rate is calculated based on individuals, right? Not like looking at germ cells. 'Cause if you were looking at germ cells, the rate I'm assuming would be much higher because you'd be capturing stuff that wasn't viable.
Great point. Yeah. Okay, so there's a bit of a history there, and it's kind of interesting, at least to me. The way this was done originally was by comparing the reference genome from chimp and the reference genome from human, figuring out, based upon essentially paleontological data, the number of differences that we observe between those two genomes and the time to most recent common ancestor. That ended up with a rate that was about twice this. And then 10 years ago, Lynn Jordy's lab collaborated with Lee Hood. He's the guy, if you remember, Lee Hood was the guy that automated Sanger sequencing with lasers. So they actually sequenced a human genome. They sequenced mom, dad, and kids and directly measured the differences that led to this rate. But to your point, the rate is probably going to be higher if you actually directly sequenced gametes. So if you sequenced a population of sperm, you probably would see a higher rate in at least some of the sperm because gametes that have a really, really high mutation rate probably more often don't lead to viable embryos. That work is really just now beginning. I'm actually writing a grant that's trying to do exactly that. There's been 2 studies in the last 2 years, but essentially what that requires is either deep sequencing of a population of sperm cells or using single-cell sequencing technologies to directly sequence an individual sperm, which is much harder. But that's, I think, really the future, and connecting that to endophenotypes like fertility and developmental phenotypes is the end goal. Right. So this paper is the one I was talking about where they directly sequenced families, and that's gave a much better estimate than this paper from 2000, which really compared the chimp and human genome. So essentially, we have a pretty good sense of what the human mutation rate is on average. It's variable from individual to individual, and they're rare. We're talking about 80 on average mutations in a genome that's 3.1 billion base pairs. However, we know the reason we care about these is that we know that most developmental disorders are caused by these germline mutations that arise spontaneously. So they're understanding which parts of the genome are susceptible to mutation in healthy individuals versus those that are not susceptible to mutation is really important to interpreting new mutations that we observe in kids with different developmental disorders. Okay, so the last thing I just want to touch upon is that we know that most of the mutations that are in our genome, new mutations, came from our dad. And that, I think, makes some sense because the male germ lineage is continually dividing to create new sperm throughout the lifetime of a man. And every cell division requires a genome duplication, genome replication. every time we replicate a genome, it's an opportunity for error. Now we have an exquisite complement of DNA damage repair enzymes whose sole job is to basically scan the genome and detect these mutations, and they do a really crazy good job if you think about how many cell divisions occur and how few mutations we actually see. But mistakes happen. So most of those come from the male lineage because of replication, but exogenous DNA damage is actually a ever more appreciated role of that increase. Yeah.
And could you, I mean, it's possible that like male gametes are more exposed to exogenous factors than female.
That's a good hypothesis, and that's, I think, bearing out that surprisingly, not much research has been done on that, but yes. And so when we look at the number of new or de novo mutations on the y-axis as a function of the age of the parent, we can see that there's this really steep increase for the male lineage. So as men get older, they contribute more and more mutations to their offspring. So it works out that, at least in our hands, for every additional year of dad's age, there's 3 additional de novo mutations passed on to their offspring on average. Yeah?
What about for polyploid?
For polyploid organisms?
No, like passing on a trisomy.
Much less frequent, and the enrichment is in the maternal lineage there. So most aneuploidies are actually coming from the maternal lineage. So this is all ascertained from neurotypical or healthy individuals. So we wouldn't see any type of that bias or that increase in the maternal lineage for this data. So the last thing I want to touch upon here is that this study didn't see much of an increase in the mutations in eggs as a function of mom's age, but subsequent studies, including our own, have seen the slope is not nearly as high as in dad, but it's about 0.4 additional mutations per mom's age. And that result, when it first came out a couple years ago, was a bit surprising, because the working hypothesis for the increase in dad's lineage was because of continued cell division, but all the eggs that a mother has are made while she's developing in her own mother's womb. So there's not this continuous cell division. So that supports the hypothesis that exogenous DNA damage is actually a contributor as well to germline mutation, because it does increase in the maternal lineage, and as mothers get older, they're probably subject to more and more exogenous mutagens. Most of the mutations that we observe are so-called transitions, so A's to G's or C's to T's, and that's mainly because of they're less deleterious to the structure of the DNA. So this has been seen over and over again. And also the C to T enrichment is also a function of that spontaneous deamination of methylated cytosines at those CG dinucleotides. Okay, so we've talked about germline mutation, the rates of germline mutation, and the fact that germline mutation, because they arise in a sperm or egg, are heritable, potentially. Most mutations that arise in sperm or egg are not inherited, right? Somatic mutation, by definition, with maybe an exception that I don't know about, there may be a weird exception here, but to my knowledge, somatic mutation are utterly not heritable, simply because they don't arise in the germline. You mutate your liver cells by having a fun weekend, or you spend too much time in the sun at the beach, and you get lots of mutation from UV or alcohol or tobacco use or whatever, but those really don't go into the next generation. However, we're interested in somatic mutation because that's a hallmark of cancer genomes, both blood cancers and solid tissue cancers, and that's primarily those mutations. Somatic mutations are essentially a required event. Cancer is a genetic disorder to lead to the instability and then transition from sort of a pre-neoplasia into cancer. But then subsequent to becoming cancer, the cells are dividing really, really quickly, right? So you're also accumulating mutation in a tumor genome simply because it's dividing at a crazy rate, and a lot of the apoptosis pathways are suppressed. Yeah.
Are there any plans or any studies right now that are longitudinal studies sequencing some of these genomes when they're younger and when they're older? Has that been done yet?
It's being proposed. There's probably active studies that are unpublished. The only study I know of that's done that is something called the Snyderome. Mike Snyder at Stanford has been studying himself for about a decade now. He has diabetes, and he's been basically taking epigenetic RNA transcription and genomic profiles of himself for a while. I haven't read those papers. It hasn't been that interesting because it's just N of 1. But I think that will be done. The hard part is consent, getting people to do that and participate in those studies. That's fairly onerous. But yeah, it'd be an interesting study. Okay, so talk a little bit. I'm going to give a crash course in selection and genetic drift, which are fundamental properties in population genetics. I think either this fall or next year, Ellen Leffler and Nathan Clark are going to be teaching a much more scaled up population genetics and evolution course. If they do, and you're interested in this stuff, take it. I'm just going to give like a 10-minute overview of some basic processes. So selection. I think everyone has at least heard of natural selection. It works in two directions. So adaptive selection is when a new mutation confers a fitness advantage in a particular environment, and as a consequence of that, that mutation rises in frequency. And then there's so-called negative selection, also known as purifying selection, which is the opposite. The mutation confers a fitness disadvantage, and therefore if that selective disadvantage is really strong, it will eliminate that allele from the population. Okay? So let's imagine we've got two sets of chromosomes. Chromosomes with a C that I'm denoting as blue circles. So you can imagine these as like marbles in a bag. And chromosomes with a T allele at this site are denoted as a red circle. So this is what the founding population looks like. There's a lot of blue, and then there's one cell with a T chromosome. If that mutation, let's say we're talking about chromosomes that exist in a group of individuals that have decided to move and live in the Himalayas 20,000 feet. And let's imagine this mutation confers an advantage at high altitude. So maybe it allows red blood cells to carry more oxygen. Over time, what we should see is that mutation should increase in frequency because individuals lacking that allele may die earlier and have less offspring. And individuals that have that mutation or that new allele will live longer, outcompete, and do better in that new environment and are therefore adaptive. So that would be a signature of positive selection. This allele confers a fitness advantage, and so it's positively selected over time. If it's deleterious, it should either be eliminated or imagine that it was higher frequency. It was like roughly equal frequency between C and T before moving up to Nepal, and then all of a sudden in that new environment, that T allele is bad news. So then over time reduces in frequency. So that'd be evidence of negative or purifying selection. However, mutations can increase, decrease, or stay the same frequency just due to random processes. Just the randomness of mating can lead to increases or decreases in the frequency of a mutation that arose in the germline that have nothing to do with selection. Yeah?
So you're saying, you brought up the example of mating, but sexual selection isn't random. So where would sexual selection fall on this?
Right. So the challenge here is what I'm trying to outline is that what we end up seeing, usually when we sequence a collection of individuals, is we measure the DNA and we get a measurement of frequency. We usually don't have these earlier time points. So what we're left to do is measure the frequency of a given mutation in a population, and we often want to compare that population, to the individuals that are living now in Nepal versus maybe people who stayed at the equator, and we're comparing the frequencies, and then we're left with a challenge of teasing out whether the apparent frequency or differences in frequencies in a mutation in one population versus the other is the result of sexual selection, adaptive selection, purifying selection, or just randomness. And it turns out that that's really a really difficult problem. People have made their entire careers on developing new statistics to try and tease that apart. But in a nutshell, there are very, very few examples in the human population of clear mutations that have had a very strong fitness advantage and led to this, what's called a sweep. This mutation sweeps in frequency. It goes from very, very low to very, very high. The few examples that I know of off the top of my head are alleles that confer fitness advantage in adapting to high altitudes. That essentially, the mechanistic reason behind that is being able to carry more oxygen on red blood cells. There's been multiple independent adaptations to mutations in lactase that allow people to process a high dairy diet better. And there are a handful of others, but most of the adaptive selection in the human population has been selecting on existing variation that exists in a population rather than some new special mutation arising and then all of a sudden that thing just takes off. And that's primarily because probably the selective pressures on human are not as extreme as on other organisms. And so there's less of a need to, or less pressure to actually drive that really rapid adaptation. Okay, so another process is a bottleneck. So let's imagine we have this mixed population. Now we've got a bunch of chromosomes that are roughly equally frequent, C's and T's. Then all of a sudden, let's say this is the population, say living in sub-Saharan Africa, and then all of a sudden there's this 5-year drought, and it's just intense. There's no rain, our food is dying off, and so a group of people decide, forget this, we're out. And they leave and they move north in search of a better environment, better food. And so there's this bottleneck of people that end up leaving. And in that bottleneck, if it's a random process of who chose, those people that chose to leave don't know whether they have a C or a T allele, right? It's just a group of people that are like, we've had enough. And in that population, it might be a very skewed ratio of red to, or T to C alleles versus what was in the larger founding population. And so bottlenecks like this can occur because of war, migration, extinction does this. Most nearly extinct species have extremely low genetic variation because of this bottlenecking process. For example, elephant seals in Baja basically have no genetic diversity. They're essentially inbred. They're all inbred individuals essentially because of bottlenecking. So over time, let's imagine this group stays in sub-Saharan Africa. This group moves up into Eurasia, and they like it there. This is much better. There's food, there's no drought, we're going to stay here. Over time, they intermate, and the population grows up, and now we have this very different allele frequency. The red is much more common in this outgroup than blue versus the founding population. So if we then, this is what happened as geneticists 2,000 years later, or 30,000 years later, more better estimate, we don't know what led. We just sequence individuals from this group, sequence individuals from that group, and we observe these differences. It could be that this allele was purified. We could hypothesize that this allele was purified in this population. It was a fitness disadvantage. we could infer that this allele, the red one, conferred a fitness advantage, and that's why it was more frequent in this subpopulation. Or we could infer that, oh, this is just randomness, this is drift. But this gets at the challenge of trying to identify which mutations or which genetic variations are under selective pressure. Because there's all these other processes, like random processes like genetic bottlenecks and drift, that can give rise to these apparent differences in allele frequencies between different populations. That has nothing to do with fitness advantages or fitness disadvantages. Just human demography and randomness. And these notions are really fundamental to understanding human evolution. So you've probably heard of the out of Africa theory. I think it's probably moved beyond theory now. The data are really strong. This is a complicated figure, but I'll explain it in words. Essentially, there have been multiple waves of migration of individuals out of sub-Saharan Africa. And these multiple waves over time explain why if we sequence individuals from, say, Southeast Asia versus people from Europe versus people from sub-Saharan Africa, we will observe genetic variation that is at very different frequencies among the same site in the genome is polymorphic, but the frequency of the different alleles is very different across those different populations. And most of the time, the vast majority of the time, that has nothing to do with selection. It has to do with the individuals that left sub-Saharan Africa and the size of the groups of individuals that went and how long ago that migration event occurred. The longer ago that it was, the more probable that either the mutation has decreased markedly versus the founding population or increased markedly versus the founding population. Both directions are equally plausible. So when we look at the same site in the genome, so right now we're looking at chromosome 5, position 25,967,094. There are two different alleles that are in the population at this SNP, single nucleotide polymorphism. There's a C allele and a T allele. The T allele is predicted to be the ancestral allele, so it's common to both chimp and human, so it's probably in our common ancestor, so it's the ancestral allele. But check this out. If you look at individuals from Western Africa, you never see that C allele. But if you did the same study and you only looked at individuals, what is that, Saudi Arabia maybe, something like that, it's like 50% allele frequency, right? So one of the realizations from this migration out of Africa theory is that to really understand genetic variation in the human population, we need to sample more than one population. So when interpreting genetic variation, say this allele C was associated with some disease phenotype, our inferences about how correlated that allele is with the disease phenotype really matter as a function of what ancestry the individuals we're studying are. So we would interpret the risk of this allele, say in people from Western Africa versus Asia, very differently, right? And that's really as a result of drift, this randomness of alleles going up and down in frequency as a function of the randomness of mating, the relative randomness of mating, and migration and bottlenecks and all these other things. So one attempt to dig into this more deeply is a project that I already talked about, this 1,000 genomes, actually 2,504 genomes project. They wanted to take a deeper dive into the genetic variation that exists among all these different populations. So each of these populations are given these 3-letter acronyms. Some individuals from Puerto Rico, they're PUR, from the Mexico-California border, MXL, Asians from southwestern United States, Yorubans, et cetera, et cetera. You can look those up if you care. The main result that I want to highlight is on the x-axis here, they've got different world populations in different colors, and they're labeled here. And then on the y-axis is the average number of polymorphic sites observed in individuals from these different world populations. And you can see there's this cluster of different world populations that have on average like 4.1 million genetic differences. And then there's this other cluster up here that's almost 20% higher, like 5 million genetic variants per genome. And it turns out that these are populations that have been from sub-Saharan Africa, or from Africa more broadly. And the observation is that those individuals from Africa have higher genetic diversity. And I think based upon this out of Africa theory, it makes sense because those populations have been less prone to bottlenecks owing to migration. So the genetic diversity that existed in those founding populations has existed and continued to flourish, whereas populations that left Africa were more prone to bottlenecks, and the genetic diversity was lost. And so this gets at one of the sort of the, I think, an important concept in human genetics. The notion of race is absolutely an artificial, human-derived concept. It is not a genetic concept. To ram it home, if you compared the genomes of any two individuals from sub-Saharan Africa, they will differ at the DNA level much more than if you compared the DNA from an individual from sub-Saharan Africa to an individual of European ancestry. So our notions of race and things that we get in ancestry that we get from 23andMe are really our societal notions of race are phenotypic and sort of stereotyped notions rather than anything that bears out at the genetic level at all. Okay, so here's the numbers. This is basically table form of the previous slide. We're not going to walk through this slide mercifully, but all I want to show you is the numbers here. People of broadly of African ancestry have 4.3 million single nucleotide polymorphisms and about 625,000 insertion deletion polymorphisms on average. And then if you compare that to Europeans, much less genetic diversity, 3.5 million on average and 546,000 indels on average. Okay. Other important concept in genetics in humans and in any species for that matter is that most of the genetic variation that exists, let's imagine we sequenced everyone on Earth, every human on Earth, and we plotted, and we made a plot like this, where on the x-axis is how frequent, for each of the variants that we identify from looking at everyone on Earth, how frequent are they among all 9 billion individuals? So if a variant is present on one chromosome out of 9 billion people times 2 chromosomes, because each of those 9 billion are diploid, it would basically have a frequency of nearly 0, because it's 1 over 18 billion. If it's very common among all those 9 billion people, it's going to be way out here, right? And what you can see is that notice on the y-axis, this is the number of variants observed among those 9 billion people at that frequency. This is on log scale. So the vast majority, way up here, of the genetic variants that were found in the 1,000 Genomes Project, so 2,504 people, but this plot would apply if we sequenced all 9 billion. The vast majority of genetic variation that we find is really private to a handful of individuals, like one individual or a couple or specific to the family. And that, I think, it's a bit of it's a confusing notion often because most of the variation is quite rare. So this disconnect between how prevalent different types of variation are across the genome versus their frequency within the population. But it's an important thing to recognize that most of the genetic variation that we observe in your genome will be it will be present in other individuals, but every new individual adds a handful of new alleles that have never been seen in anyone else on Earth. Okay. Yeah.
How do you interpret, like, past the 0.9, that little increase?
Yeah, I'm glad you asked. So there was a question from the back. Well, probably people aren't sitting in the same spot anymore, but a few classes ago there was a question about the number of individuals that went into forming the reference genome and how a consensus was determined. So it's just a handful of individuals that form the reference genome, and a consensus was chosen to form that sequence from those individuals. And most of the time, the allele that is in the reference genome is the more common allele. And so everything that we observe by sequence 9 billion genomes is less common than what's in the reference genome. This is the exception. These are cases where the reference allele is actually the minor, the less frequent allele. And what we observe by sequencing 2,504 individuals is that there's looks like about 10 or 50,000 variants throughout the genome, where what's observed among those 2,500 individuals is different than the reference allele. Okay, so in the last 10 minutes, I just want to cover a little bit about haplotypes. So everything we've talked about so far is genetic variation at a single site on a single chromosome. But obviously we inherit a whole block, a continuous haplotype or gamete from dad, a continuous gamete from mom for each of the chromosomes. And those haplotypes that we inherit from mom and dad are a combination of different alleles at all the polymorphic sites in the human genome. So let's say this is my genome. Green is what I inherited from dad. That's my paternal haplotype. Blue is what I inherited from mom, my maternal haplotype. In each of these little lollipops here are sites. We're just looking at a small snippet of the chromosome. Sites that were inherited together. So this G allele was on the same haplotype as G and T here, whereas on dad I got AGC. So I'm heterozygous at each of these three different sites, but typically we're just looking at a particular slice in the genome, and we recognize, oh, I'm heterozygous AG. What we really care about is the full complement of alleles that I inherited on one haplotype and the other, and that's much more powerful information to connect haplotypes with traits and disease phenotypes. So, and we know, let's say this is my genome, paternal and maternal haplotype. Every gamete that I produce, or any individual produces, is a recombinant. The recombination occurs during meiosis, so there's additional shuffling. So there's new mutation that arises in gametes, but we also get additional genetic diversity because recombination occurs, right? So maybe there's a crossover event here that leads to a new gamete, new haplotype that has never been seen before, which is a mixture of the two alleles, the two haplotypes that I inherited from my parents. So now this is a gamete that is G crossover GC, so a mixture of the two alleles. Okay. So, and as we know from probably genetics that everyone has had in undergrad, that this frequency of recombination is a function of how far apart those two markers are. So, I mean, I think it just follows naturally. If this site and this site are really far apart on a chromosome, there's a lot more genomic real estate within which recombination can occur than two markers that are really close to each other. So it's more likely that a recombination event will occur between here and here than between G and C. And this gets all the way back to work that was done in T.H. Morgan's lab way back when. And so this is typically measured. This terminology isn't used all that much in human genetics because we have a physical map of the human genome. We have actual base pairs and positions, a coordinate system. But prior to that, we talked about the distance between markers in a genome as a function of centimorgans. This gets back, the term comes from T.H. Morgan's lab. So essentially, 1 centimorgan is the equivalent of a recombination frequency of 1%. So 1% of gametes will be recombined between these two markers, say, and so they have a distance of 1 centimorgan. On average, a centimorgan works out to about 1 million base pairs, but that's highly variable. That depends upon how the recombination rate between two markers. But the main reason I want to bring this up is that when we sample and when we sequence an individual's genome, we're not only finding new variants that exist in that individual's genome, but we're also getting a mosaic of that individual's parents' genomes. And that type of data has been used to figure out essentially a recombination map for the human genome. This has been done in Drosophila and in mouse, and that's really important for looking at if you've ever done a linkage study or know about a linkage study. Essentially, you're using recombination or lack of recombination to track the segregation of alleles with a disease trait. Okay, so with that, we'll stop here. In the next lecture, we'll start talking about modern DNA sequencing technologies and how we can use them. And I think we'll introduce the second hideous file format in genomics called the FASTQ format. Just a reminder that second homework is due Thursday. We're going to be starting to grade homework 1 this week. I need to turn on the permissions for you to upload your homework for homework 2. I'll do that. And are there anyone having problems that we should address? We have a couple minutes. Yeah.
I just have a question. If
Yeah.
we're working on the homework,
Yeah.
we're looking around online, and we see a neat command that can help us, but we haven't covered it in class. Can we try it out?
You can. You shouldn't need to. It would be better to, unless I'm mistaken, all the questions can be answered with the same commands that we've covered in class, perhaps with options that haven't been covered in class, but it shouldn't be any new commands. Yeah.
Is there a good source for explaining like all of the different, what a GTF file, like all the different components, like, especially like column 9 that has so many?
Yep. If you just Google GTF format.
Well, I mean, I've done that, so I was hoping that you'd have something.
Well, GTF format and Google Ensemble without an E at the end. That's the standard. Yeah. Oh, sorry. Yes. There's a bonus question for homework 2. That is an exception. That might require a command that we didn't cover. So that brings up a broader point that I wanted to emphasize earlier. And I may have, but I'll say it again. Pretty much everyone that does this type of work day in, day out, Googles stuff all the time. Stack Overflow, there's a website called BioStars. No one memorizes this stuff. So just get used to Googling. Part of what I hope comes out of this class is that you know what to Google when you go back to your labs and are doing research in this area. We're not going to learn everything that there is to know. You need to depend upon your Googling skills and other resources. And chances are, if you're facing a problem with any of these formats or trying to do something, someone else has faced the same problem before and has answered it. So I still do that stuff all the time. Everyone in my lab does. You should too. Okay? Thanks.